Hey, my name is Deyang. Today I'll be presenting on my poster, which is investigating the STAT1 associated immune checkpoint gene expression and spatial profiles of immune cells in high-grade serous ovarian cancer, or HGSC. Over 16,000 women die from HGSC each year in North America. While these patients initially respond to chemotherapy, the majority exhibit acquired chemo resistance and have not yet benefited from more recently developed immune checkpoint blockade or ICB therapies. Previous investigations in our cohort of patients saw that high interferon-induced STAT1 and also high CD8 positive T cells within tumors was associated with improved survival and better response to chemotherapy. This is in contrast to immunologically colder tumors, which have less expression of interferon-associated genes, lower density of CD8 positive T cells, and often respond poor to chemotherapy and immune checkpoint blockade. However, while increased STAT1 expression and CD8 positive T cells within tumors is associated with an improved prognosis, many of these patients unfortunately still exhibit acquired chemo resistance. And so establishing the factors underlying differential STAT1 expression is therefore critical to the rational use of immune therapies in HGSC. Our objective was to determine STAT1 expression associated whole transcriptomic alterations and immune cell distributions. And in terms of approaching this, we had a discovery cohort that was stratified into STAT1 high and low based on protein levels of STAT1 in these tumors. These tumors underwent RNA sequencing, and then that data was used for differential expression analysis and also cybersort-based immune cell profiling. And all findings were validated in a validation cohort that was using the TCGA data. That's available online. And then we also did multiplex immunohistochemistry training for various immune cell like CD8, CD68 macrophage, and also immune checkpoint markers like IDO or PDL1. So here we can see that in between STAT1 high and low tumors, there was quite a bit of differential gene expression here, as evidenced in this heat map and these volcano plots. We found that in STAT1 high tumors, there was significantly increased expression of immune checkpoint, interferon, and MHC-associated genes, as well as looking at the cybersort-based in silico immune profile, and we saw that there was also higher M1 macrophage signature and also CD8 T cell signature in the STAT1 high tumors. Here are some of our representative images for our multiplex immunohistochemistry, and this data has been quantified and validated, and it's currently undergoing statistical analysis. So overall findings from the study will provide further evidence for the complex roles of STAT1 in the HGSC tumor immune microenvironment. Increased co-expression of multiple immune checkpoints in STAT1 high HGSC tumors could contribute to potential adaptive immune resistance. These findings will potentially inform rationalized immunotherapy trials in HGSC.